The overall goal of this protocol is to identify novel ice-binding proteins, or IBPs, from plants by testing for ice-binding activity and isolating native peptides using ice-affinity purification.
This method can help answer key questions in the field of plant freeze tolerance, specifically ice-binding proteins. The main advantage of this technique is that it allows you to isolate ice-binding proteins from native extracts without the use of complicated purification methods.
Visual demonstration of this method is critical, since the apparatus setup can be difficult to conceptualize.
Begin this protocol with splat apparatus setup as described in the text protocol. To induce the expression of ice-binding proteins, or IBPs, in freeze-tolerant grasses or other freeze-tolerant plant species, cold acclimate plants for up to 1 week at 4 degrees Celsius under low light conditions. Obtain approximately 0.1 gram of leaf tissue by cutting at the base of the stem and then placing in a 1.5 milliliter tube. Immediately flash freeze the sample in liquid nitrogen and store at -80 degrees Celsius until use. To lyse the cells, grind the tissue sample into a fine powder using a mortar and pestle under liquid nitrogen. Ensure that the liquid nitrogen does not evaporate during the homogenization process. Immediately place the ground samples on ice and allow the liquid nitrogen to completely evaporate. Then, add 1 milliliter of a native protein extraction buffer and pipette to mix. Gently shake the samples overnight at 4